Good day, everyone. I am Sir Roy, and for today's lesson, we're going to talk about non-Mendelian inheritance. But before that, let's have first a quick review. Inheritance falls under the topic of genetics, or the study of heredity. When we speak of heredity, we are referring to the passing on of traits from parents to their offspring. Traits are the characteristics. The man behind genetics is none other than Gregor Mendel. He is considered to be the father of genetics, an Austrian monk who studied heredity in plants. Let us also have the following terms. First one, we have the trait, an inherited characteristic of an organism. The trait could either be dominant or recessive. When we speak of dominant trait, an inherited trait that is present even when inherited only from one parent. A dominant trait is always written in capital letter. On the other hand, when we speak of recessive trait, the form of gene that shows up only when inherited from both parents. It is usually written in small letter. The next term is heterozygous. These are two different genes from the same trait. For example, we have a combination of a capital letter and small letter. And we have the term homozygous, the opposite of heterozygous. It means two identical genes for the same trait. For example, we have here a homozygous capital letters and the other one is homozygous small letters or written in lowercase. For the genotype, we are referring here to the letter combination that makes up a trait. So these are the given examples. These are the letters that represents the trait of the parent or the offspring. For the phenotype, the physical characteristics of a trait. So the phenotype tells us about the characteristic which is shown by the genotype. And the last one is the Punnett Square, a tool used to predict the possible breeding outcomes. So how are we going to do the Punnett Square? Let us follow the following step-by-step procedure. For the first step, draw a 2 by 2 square. Draw a box and divide it into 4 smaller squares, just like shown in this picture. For step 2, name the alleles involved. Choose a letter to represent the alleles. So as you can see, you have to write the dominant allele with any capital letter and the recessive allele with the same letter in lowercase or in small letter. For step number 3, check the parents' genotypes. So each parent has two alleles for the trait, just like every sexual organism, so their genotype will be two letters long. Could either be heterozygous, consists of one uppercase and one lowercase. Homozygous dominant could either be consist of two capital letters or two uppercase. And we have here homozygous recessive or consists of two lowercase letters. For step number 4, label the columns with the parent's genotype. So the genotype of the parent could be written in either ways. For step number 5, have each box inherit the letters from its row and columns. So you have to distribute the letters on their particular or on their designated portion in the box. So that is how we do the Punnett Square. Let us now proceed to our main topic, which is all about non-Mendelian inheritance. These are the rules of inheritance which does not follow the Gregor's Mendel's laws. It refers to any pattern of inheritance in which traits do not segregate in accordance with Mendel's laws. The first one is what we call as the incomplete dominance. The pattern of heredity in which one allele is not completely dominant over another. Just like shown in the picture, as you can see, if the parent are the red flower and the white flower, the resulting offspring would be pink flower. What does it mean? It means here that one allele is not completely dominant with another allele. Let us use the example of snapdragons. If we're going to combine the red snapdragons and white snapdragons, most probably the resulting offspring would be 100% pink snapdragons. How does this happen? Let us prove how the following flower color was produced in the F1 and F2 generation. So here, the parent would be the red and white flower snapdragons. For the first generation, if we're going to cross red and white flowers, the resulting color would be 100% pink. Now, for the F2 generation, the resulting flower color would be 25% red, 50% pink, and 25% white. So let us use the following legends. For red flower, we have FR. For white flower, we have FW. And for pink flower, we have FR-FW. As a reminder, for F1, we have to cross the parent and another parent. For F2, we have offspring to offspring. Let us have this example. What if we're going to cross a red and white snapdragons? So the first step, you have to combine the two genotypes which are encircled. So we have FR and FW. Don't be confused. Let us follow the alphabetical order. For the second one, combine this portion, FR and FW, and write it on the designated area. For the third one, let us combine the encircled genotype. So we have here FR and FW again. And for the last step, we have to combine the last pair, FR and FW. So as a result, we have here the result of the Punnett Square. So our genotype is, we have 4FR-FW. This is our genotype. And what would be our phenotype? What does it mean by FR-FW? We have to look again to our legend. So what color is represented by FR-FW? We are referring to pink snapdragons. So we have the phenotype of 4 pink snapdragons flowers. Next, for the codominance, the pattern of heredity in which both alleles are simultaneously expressed in heterozygote. Example of this would be our blood types. So given a table would be the phenotype and the genotype of our blood type. So we have here blood type A, IA, and IA. So this one is a homozygous blood type A, and the other example is IAI. This is an heterozygous blood type A. For blood type B, we have IBIB. This is a homozygous blood type B. And for heterozygous blood type B, we have IBI. For blood type AB, we have IAIB. And for blood type O, we have here two small letter I or two recessive I. Let us have this example. Mom has type A blood. Let's say this is an homozygous one. Dad has type AB blood. What possible blood types could their children inherit? Let us have this table. For the parent, we have mom and dad. For the phenotype, the blood type of mom is type A blood. Let's say it is homozygous. That's why it's IAIA. For, for dad, we have type AB blood. So that would have a genotype of IAIB. So again, we use the table as a pattern for our genotype. Next, let us perform the Punnett Square. So we are going to cross the blood type A with blood type B, AB, typographical error. So we have IAIA and IAIB. So up here, the first one, if we're going to combine the two IA, so we have IAIA and IAIA. And the other one, we have IAIB and IAIB. So the resulting genotype would be 2IA-IA and 2IA-IB. So what does it mean? We have here two blood type A and two blood type AB. Where did we get the phenotype? Again, we base it from the given table or legend. The next one and the last type of non-Mendelian inheritance is the multiple allele, a gene that is controlled by more than two alleles. Example of this one is the genotype of rabbits. We have here the colors of black, chinchilla, Himalayan, and albino. So at the upper portion, you would see the genotype of the following rabbits. What if we're going to cross a black rabbit and a chinchilla? So we use the following genotype based on the table that I've shown a while ago. So if we're going to do the Punnett Square, so this is the result of the first combination, the second combination, the third, and the fourth. So as you can see, if we're going to interpret the Punnett Square, the genotype would be 4CCCH. It has a phenotype of 4 chinchilla. Where did we get this chinchilla? We base it again from our table or legend. So that is what we called as the multiple alleles. Thank you for watching.